Hi there, my name is Stuart Harper. I'm a senior at the University of Washington in Seattle studying neuroscience, and I'm really excited to present to you today the work that I've been doing in Professor Hanali Rohola Baker's lab at the Institute for Stem Cell and Regenerative Medicine. Diffuse intrinsic pontine glioma, or DIPG, is a rare form of pediatric brainstem tumor. The average age of diagnosis is between 5 and 9 years old, and there are approximately 300 children per year that are diagnosed with DIPG in the United States. There is currently no effective treatment for DIPG. Surgery specifically is ineffective because of the diffuse nature of the cancer within the pons, where the cancer cells form a network around the healthy pontine cells, making surgery impossible. Chemotherapy is also ineffective, and so the current standard of care for these patients is a palliative radiation therapy, which helps to alleviate symptoms for around 2 months before they recur. Because there is no effective treatment for DIPG, no patients will recover following a DIPG diagnosis. The average survival duration is only 10 months, and only 1% of patients will survive 5 years following their diagnosis. This is in stark contrast to many other forms of pediatric cancer, which have seen the 5-year survival rates increase drastically over the past 50 years. To better understand why DIPG is so resistant to standard cancer treatments, the field has taken a genetic approach to identify mutations which can better characterize DIPG. Around 80% of DIPG patients have a mutation in the histone 3 isoform H3.3, which changes the lysine 27 to a methionine. H3K27 methylation is not possible on these mutated histone 3.3 isoforms, which drastically changes the epigenetic landscape of these cells. Analysis of the H3K27 methylation landscape shows a global reduction in methylation compared to healthy glial cells. DIPG cells also show hypermethylation spikes in conserved regions. Because H3K27 trimethylation is a repressive epigenetic mark, addition of methyl groups serve to repress transcription at local sites. Therefore, we anticipate these hypermethylation spikes to be regions of extreme transcriptional repression. One gene which is located within a hypermethylation spike is the tumor suppressor protein P16. P16 helps to regulate the transition from G1 to S phase in the cell cycle by inhibiting the interaction of cyclin and CDK. A repression of P16 would allow for aberrant activation of the transcription factor E2F, allowing these cells to easily transition into S phase. And so because of the presence of a hypermethylation spike on the P16 gene, we hypothesize that the repression of P16 transcription in DIPG cells is playing a role in the tumorigenesis of DIPG. Shifting gears slightly, the polycomb repressive complex 2 is the complex responsible for H3K27 trimethylation. There are 2 important subunits I want to point out. EZH2, which is the catalytic subunit that actually adds methyl marks, and EED, which is critical for its interaction with EZH2, other PRC2 subunits, and its identification of trimethyl marks. Overall, the function of PRC2 is the addition of methyl marks on H3K27, serving to repress transcription of local genes. Now to bring in the computationally designed protein. The EED binder protein is capable of binding to EED at a much higher affinity than EZH2, effectively displacing the catalytic domain of PRC2 away from the complex and blocking the addition of methyl marks. To allow for precision targeting of EB to a specific gene, EB was fused to DEDCAS9, which itself can integrate a guide RNA of our choosing. This allows the complex to displace EZH2 at a location of our choosing, blocking methylation and allowing transcription of that specific gene to remain on. So to bring all of this together, I told you that P16 transcription is repressed in DIPG cells given the hypermethylation spike residing near the P16 gene, which we hypothesize contributes to DIPG tumorigenesis. By targeting EB-DEDCAS9 to the P16 gene using a P16-specific guide RNA, we are aiming to remove the hypermethylation to restore P16 expression in DIPG cells, with the ultimate goal of reducing DIPG viability. Here's a cartoon of the EB-DEDCAS9 plasmid, which contains the EED binder protein fused to DEDCAS9. The plasmid also contains an M-cherry insert, allowing us to visually confirm if the cells have taken in the plasmid. It also contains the TET-on promoter, which allows us to promote transcription of both EB-DEDCAS9 and M-cherry inside our DIPG cells by adding doxycycline. Our next step was to generate a p16 specific guide RNA, which was capable of targeting EB-DEDCAS9 to the p16 gene and increasing its transcription. Here we see the transcription start site for p16, and to the right is the region upstream of TSS. You can see the varied locations of the p16 guide RNAs that we generated, some near TSS, some just upstream of TSS, and some far upstream of TSS. We tested each of these p16 guide RNAs in iPSCs and found that guide 1 was the most effective in increasing p16 expression. So for the rest of the data, the guide RNA that we have been using is guide 1, located just upstream of TSS. The next step was to optimize the transfection protocol for these DIPG cells, which is something we're still working on perfecting. The current protocol transfects both the plasmid and guide RNA using electroporation into approximately 2 million DIPG cells. I add doxycycline at various time points to increase transcription of EB-DEDCAS9 before harvesting on day 7 for qPCR analysis. Here you can see the normal morphology of these DIPG cells, which grow in these neurosphere clusters. This image was taken 7 days following transfection, so right before we harvested the cells, and you can see M-cherry expression, indicating that the transfection was successful and that these cells are expressing EB-DEDCAS9. And here's some of our most exciting data from these DIPG cells, where we see a 19-fold increase in p16 RNA in DIPG cells which had been transfected with both the EB-DEDCAS9 plasmid as well as guide RNA, compared to DIPG cells which had been transfected with the EB-DEDCAS9 plasmid, but not with the guide RNA. I told you at the beginning of this presentation that around 80% of DIPG cancers contain this histone 3 mutation, changing the epigenetic landscape of H3K27 trimethylation. Specifically, I showed you the hypermethylation spikes, which serve as extreme transcriptional repressors. The goal of this project is to generate a p16-specific guide RNA, which is capable of integrating with EB-DEDCAS9 to increase p16 transcription in our DIPG cells. By using this precision targeting, we hope to reduce DIPG tumor viability without alternative off-target effects, which might otherwise be possible in a global repression of PRC2 activity. Although we are still working to optimize our transfection procedure, there are a number of next steps that I'm looking forward to exploring. I next plan to test the viability of these DIPG cells following p16 restoration and determine if these cells can continue to divide even when p16 is restored. Other long-term projects include an in vivo application, such as a mouse model, or a brainstem organoid. And last, I mentioned before that these hypermethylation spikes occur across the genome in DIPG cells, so I anticipate that there are other important cell cycle regulators which might also be dysregulated in DIPG cells because of the histone 3.3 mutation. Identifying other candidate genes and examining their effect on DIPG viability is another potential path for this project that I find really interesting. I want to give a special thank you to the Mary Gates Endowment, specifically for the research scholarship they awarded me for the past 2 quarters. Thank you so much for helping to fund my work. I also want to say thank you to my 2 mentors on this project, Professor Rohola Baker and Ashish Fall. Another thank you to Dr. Shiri Levy for her help with EB-DEDCAS9 and the rest of the Rohola Baker Lab for their continued support and encouragement. Thanks so much for listening, and I'd be happy to answer any questions.